All right, everybody, welcome back to part two of chapter three. I'm picking back up on slide number 23, and we will start by talking about movement through the cell membrane. So, big category wise, this is broken into passive transport and active transport. The big difference between the two is that passive will require no energy, while active will require energy, typically in the form of ATP. So we'll talk about the passive transport methods first. So all of these next few are passive transport. Okay, diffusion. So this is a good example to illustrate diffusion. So imagine you drop a dye tablet into a beaker. So initially, all the color or particles of the tablet sit right by where we dropped it. And over time, we'll see that they spread out relatively evenly in the water. So it's moving from an area of high concentration to an area of low concentration. And eventually it kind of all evens out where it'll be evenly distributed. And here's just a little bit of an animation of this example. So as we said, we're going to move from an area of high concentration to low concentration. So the difference in concentrations is known as the concentration gradient. So when particles move from an area of greater to lesser concentration, it's moving down the concentration gradient. So think high to low, it's going down the concentration gradient. Okay, osmosis is a similar concept to diffusion. The big difference is that this time, rather than particles doing the movement, you're going to see water do the movement. So there's also a membrane involved. So think of your cell membrane. So in this case, we have a 5% solution on one side and a 10% solution on the other side. So side B has twice as much albumin as side A. So in order to even that out, rather than particles moving through the membrane to even it out, we have water moving from the side of lower concentration to higher concentration to even it out. So as the 5% side loses water, it becomes 7.5%. As the 10% side gains water, it becomes 7.5%. So this difference in water now creates what's known as an osmotic pressure. So there's once again a concentration gradient to start out with, but this time rather than the particles spreading down that concentration gradient, you're going to see water moving from an area of actually lower concentration to the area of higher concentration. So more water now creates osmotic pressure between the two sides. Again, there's an animation of this. I think I would definitely watch that. Osmosis is not always the most easy concept, but it's a very important process within the body. Okay, tonicity is the ability of a solution to affect fluid volume and pressure through osmosis. So tonicity is, when we speak of tonicity, we're talking about osmosis as well. So there's a few different types of tonicity. So isotonic is something with the same concentration of solutes that are inside the cell. So everybody's happy. We have, say, for that last note, we maybe have 5% albumin outside the cell. We have 5% albumin inside the cell. We don't need any movement of that water because we're already balanced. Hypertonic, you're going to have a higher concentration of solutes outside of the cell compared to inside the cell. So because there's a higher concentration of solutes outside the cell than inside the cell, water is going to move out of the cell into the outside environment in an effort to balance that out. On the flip side, you could have a hypotonic solution. So that's going to be a solution with lower concentration outside the cell as inside the cell. In this case, water is going to flow into the cell to try to balance out that concentration. Again, I put a link down for an animation that illustrates those concepts again. They also show a red blood cell example on page 44 that I think illustrates this concept well. All right, filtration. So this happens quite often, or the best example within the body would be the capillaries. So again, this is going to occur due to a difference in pressure rather than a difference in concentration. So this is going to be from a higher to lower pressure. So water and the dissolved particles within the bloodstream in this example are going to be forced out of the capillaries into the body because there's a much higher hydrostatic pressure in the capillaries than there is outside of the capillaries. So that's how our capillaries force water and nutrients into our tissues and fluids. Another example within the body would be the kidneys, and it forces waste products out of the blood using that high pressure. So remember, this is due to differences in pressure. We're not talking about that concentration gradient concept. And you're going to see a movement from higher to lower pressure. So high pressure forces the water and nutrients to the lower pressure areas. All right, review question for our passive transport topics. So osmotic pressure is A, the force driving osmosis. Is it B, the force that drives solutes through the capillary walls? Is it C, pressure that aids venous return? Or is it D, water pressure that develops from osmosis? Take a second to think about it, pause it if you need to. And your correct answer is D. That's the water pressure that develops from osmosis. So think back to our 5%, 10% example with the membrane in between. You see the water moves from the 5% side to the 10% side. So even though the concentrations are balanced, we now have a different volume of water on either side of the membrane. And so that creates the osmotic pressure, the water pressure, differences in water. So osmosis itself is driven by the concentration gradient, not the osmotic pressure. Hydrostatic pressure is what we just discussed about with the capillaries. And then osmotic pressure really plays no role in venous return. All right, our last passive transport example is facilitated diffusion. It is the exact same concept as diffusion. So we have solutes moving from an area of high concentration to low concentration. The only difference in this example is the fact that it's using a channel protein. So this may be something that's a much larger molecule or a polar molecule that can't just diffuse through the membrane, such as glucose. Glucose is a good example of this. So when that solute attaches to the channel protein, that channel protein then changes shape to fit it into the cell itself. But even though we're using a channel protein, we are not using energy or ATP for that protein to change shapes and transport the solute from outside the cell to inside the cell. It's all moving based on that concentration gradient. So this is still a passive transport. All right, we're now going to talk about active transport. So remember, it's going to require energy. This is typically a pump or a vesicle. Okay, the most important pump or the most talked about pump in the body is probably the sodium-potassium pump. And you'll hear this most often discussed when it comes to maintaining the electrical potential energy that your nerves utilize to send their signals. So the little red dots are your sodiums and the green squares are your potassiums. So what ends up happening is that 3 sodiums from inside the cell get pumped out of the cell using ATP, and it brings back in 2 potassiums. And there's an animation that goes with this as well. We're about to go over the steps on the next slide. Okay, so as I said, you have 3 sodiums, and they're going to be inside the cell. They go into their receptor sites on their channel protein. When all 3 attach, that protein knows, all right, it's time to pump that out, but I need ATP to be able to do that. So the sodium will end up in the extracellular fluid outside of the cell. And so the reason it needs energy or ATP to do this is because there's already a higher concentration of sodium outside of that cell. So it's actually pumping it against the concentration gradient, or we're going up the concentration. So we're going from lower concentration to higher concentration rather than higher to lower, as we've seen in our passive transport examples. So outside the cell, we get two potassiums that come into that channel protein, and that's going to go back into the cell. And so the end result of the sodium potassium pump is that two extra potassiums end up inside the cell, with three sodiums ending up outside the cell. It keeps the concentration of potassium higher in the cell, the concentration of sodium lower in the cell. So that creates some electric potential energy. It can also help generate heat. But really, like I said, what you'll see this most often referred to is the electric potential energy within the nervous system. Okay, transport by vesicles is another form of active transport. And remember, earlier in this PowerPoint, we talked about vesicles in reference to the Golgi apparatus when it's trying to ship proteins out of the cell. So cells can move larger particles or high volume through vesicles. So this can go both ways. It can go out of the cell or into the cell. So endocytosis is bringing substance into the cell. So endo means in. There's two different ways they can do this. Phagocytosis means the cell is eating something or pulling it in. Penocytosis is a liquid, so the cell's drinking what it's bringing in. Exo means to exit, so that's getting stuff outside of the cell. So when the Golgi apparatus packages and delivers a protein to the cell membrane and it exits, that would be an exocytosis. So vesicles are going to release substances to the outside of the cell. There's just a couple YouTube videos here that will show you examples of how vesicles work. All right, next question. Which of the following is a key function of the sodium-potassium pump? A, movement of large particles into the cell across the plasma membrane. B, provision of electrical potential necessary for nervous system activity. C, release of substances outside the cell. And D, provision of nutrients to the body's cells. So once again, pause it if you need to. Think about this one. The answer is B. It's provision of electric potential energy necessary for nervous system activity. I know I said that a few times, so I hope you guys got that one. So remember, endocytosis is movement of large particles into the cell. Exocytosis uses vesicles to release substances outside of the cell. And filtration is the method by which the body's cells receive nutrients. So filtration, that would be exocytosis. That's typically going to be done by a vesicle. And this also could be done by a vesicle and or channel proteins. All right, now we're going to discuss DNA. So we're all done with the transport side of things. So DNA is our genetic material. I'm sure most of you guys know that already. And it's the complete set of genes and genetic material which makes up our genome. That's all the DNA within the body. So the way DNA is put together, it's considered a double helix structure. So that's what this kind of spiral is referred to. You'll often see it organized like this in examples. So you have the backbone of DNA, or not the rungs, but the legs of your ladder. That's going to be an alternating sugar and phosphate group. So those make up the backbone. And then you have the nitrogen bases or nucleotides that make up your rungs of the ladder. So these nucleotides or bases have pairs with one another. So the four are adenine, thymine, guanine, and cytosine. Adenine is always going to pair with thymine, always. So you'll never see an A and a G or an A and a C or a T and a G or T. So you'll see here AT, 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 TA, always together. Same thing for guanine and cytosine. You'll always see C and G together. And we also have RNA. And so this is a single strand versus double. So it's just that single, not a double helix structure. The reason it's called RNA is because it has ribose sugar instead of deoxyribose. So that's the difference between the R and the D there. And then one of the other big differences that you'll see is that rather than having thymine, it has uracil. So you'll still see G and C together, but now we have A and U together when it comes to RNA. And so the DNA is housed within the nucleus, and that's kind of like our master copy. That's got all the information that our cell could possibly need. And so that DNA is pretty large in considering, I mean, relatively. So it's too big to just exit the nucleus and tell everything what to do. So it stays put. There's a few different types of RNA, the most important for us being mRNA, which is messenger RNA, and tRNA. And so the RNA is small enough that it can exit the nucleus and head to different parts of the cell to tell it what proteins and structures to make. And we'll go through that process is known as transcription and translation. That's how we make proteins, and so that's what we'll discuss next. So in general, when we're talking about DNA and RNA, I want you guys to focus on kind of what the differences between the two are, and they're two different roles. So think DNA is our master copy. DNA is the end all be all. It's got all the information our cell needs. The RNA is how we can actually utilize that information for the cell to make proteins. Okay, the first part of protein synthesis is transcription. So this is how DNA and RNA work together. So first step, that DNA or that double helix is going to unwind so that we get a single strand versus a double strand. So the RNA is, in this case, mRNA is going to make an exact copy of the opposite side of the DNA. So you see it unwinds. We have our mRNA is going to start attaching its complementary nucleotides. So like we said, you still see G and C together. But rather than T, when we have an A on the DNA, we're going to see a U or the uracil of the RNA bind with it. When we get a T on that single strand, you'll still see an adenine or an A bind to it. So the nucleotides attached, that's going to form the mRNA strand. And then three bases put together. So 1, 2, 3, that's considered a triplet or a codon. And so that gives the code for one amino acid. So one building block of a protein comes from 3 bases. So once they get its proper sequence encoding so that it has the copy, it's then going to move out of the nucleus to the ribosomes, and we will go into translation where the protein is actually synthesized. This YouTube video shows these steps of transcription again. Okay, translation occurs within the ribosome. And so we have the mRNA arrives to the ribosome. There's going to be an anti-codon that attaches to the codon. So remember, 3 bases make up a codon. So we get the match here, and that gives us the correct amino acid. Within the ribosome, these amino acids are then going to link together with peptide bonds, and depending on kind of what the code says, that's how the ribosome will know how many and which amino acids to link together to form the protein. So it may be a few, it may be thousands. Okay, so in the cytoplasm, the mRNA attaches to the ribosome. There's tRNA that is waiting in the cytoplasm that has that anticodon that we just talked about to match with the codon. So 3 and 3 match, that creates one amino acid. Then we attach the amino acids with the peptide bonds, and that's how a protein molecule is completed. I would highly, highly, highly recommend watching the videos that are embedded within here. Use those links. It's a lot easier to see it in action and match it up kind of with what we just discussed. This is a little bit of a tricky concept, and it's a super important concept for how the body works. So please take the time to watch the videos that are linked within the PowerPoint. All right, which statement about DNA is correct? It stores all of the cell's genetic information. It contains the sugar ribose. It contains the 4 bases adenine, cytosine, guanine, and uracil, or it exists as a single strand. So A, all the cell's genetic information. So the rest of those answers all apply to RNA. So DNA has the sugar deoxyribose rather than ribose. Its 4 bases are adenine, cytosine, guanine, and thymine. So remember, the only difference in terms of the bases with RNA is that there is uracil instead of thymine. And DNA exists as a double strand. So that's that double helix structure for DNA. RNA will only be single-stranded. All right, moving on now to the cell cycle. So this is broken down into 4, kind of 5 stages. Mostly these 4 stages we think about. So we'll discuss all 4 of these phases, and then we'll talk more in depth about mitosis in the mitotic phase, because that's where cell division actually occurs. And so for cells to grow, they have to divide, and this whole process is how they do that. So outside of this mitotic phase or M phase, the rest of it is all referred to as interphase. So G1, we're going to talk about this piece of the pie here. So the cell performs the tasks which it was created while accumulating all the materials needed to replicate its DNA. So it's gathering up all of the stuff that it needs to make up the RNA so that it can actually start its protein synthesis. Then you get to S phase. So the cell makes or synthesizes an extra set of DNA. So this is a little different than mRNA and tRNA. It's actually preparing to have a second set of DNA itself, because the new cell is going to need its own master copy. You don't have to worry about exactly how that happens. You just need to know that an extra set is made in the synthesis phase. We then move on to G2, which is our second gap phase. So it's going to make all the final preparations it needs prior to actually moving on to mitotic phase so that it can divide. In mitotic phase or mitosis, cell division actually occurs. As we said, everything but this mitotic phase is known as interphase. And then most of the time, this is kind of a continuous cycle. Kind of once mitosis happens, we go back to G1 and the preparation happens again, because it's going to continue to divide. In some cases, you'll enter this G0 phase, which is kind of like an on-deck circle. It's the waiting phase. And so they don't divide. And depending on the cell, this may last a day, it may last years, it may never divide again. But I really want you guys to just get a general idea for what happens in these 4. Okay, mitosis itself is that cell division. So That consists of prophase, metaphase, anaphase, and telophase. And so we'll go into those each in depth here. All right, so I do not want you guys to memorize this slide word for word by any means. What I really want is you to just have a general idea of kind of the major milestone that happens in each phase. So as we go through it, I'll tell you kind of what I want you to know from each phase for sure. So prophase, the chromatin, those little threads that we talked about that are within the nucleoplasm, we talked about that back when we were discussing the nucleus. Those finally condense and coil up, and they form your chromosomes. So each chromosome has two strands. Each strand itself is known as a chromatid. And this is going to have a single molecule of DNA, each chromatid. So, each chromatid. So the two chromatids join in the middle, and that's known as a centromere. So that's the center point. We talked about the centrioles earlier in the chapter. And so those are going to move to opposite sides of the cell. The nuclear envelope that's around the nucleus dissolves, and then there's what is known as spindle fibers forming in the cytoplasm. And those spindle fibers are used later down the line here. So for prophase, what I want you to know is that chromatin coils and forms chromosomes. That's the big one. So prophase, the chromosomes are formed. And remember, the centrioles move to opposite sides. Okay, in metaphase, you see some of those spindle fibers begin to attach to one side of the chromosome, and you'll see that chromosomes start lining up along the center of the cell. So metaphase is a little less involved. That's an easy one to remember. You then move on to anaphase. So the centromeres, remember, that middle spot are then going to divide. Now you have two chromosomes rather than a pair that are attached. The spindle fibers then pull away the newly formed chromosomes to opposite sides of the cell. So the big thing to remember in anaphase is that this is where the separation of the chromosomes or the centromeres occur. The final phase is telophase. You'll see a new nuclear envelope develop around each set of the daughter chromosomes, and then those spindle fibers disappear. So the big thing to remember about telophase is that new nuclear envelope develops. And so you now have kind of two different cells in a sense. Once the cytoplasm actually divides, then you'll truly have two identical daughter cells. They're the same as each other. So this link here will illustrate that. And if you see the pictures, that's a good way to think about it too. So let's go through that one more time. So in prophase, you'll see the chromatin finally form the chromosomes. They'll attach with the centromeres, Metaphase, you'll see the spindle fibers attached, and they all line up in the middle. In anaphase, they're getting pulled apart. That centromere breaks, and you have two different sets getting pulled to opposite sides. A new nuclear envelope will develop around each set, and the cytoplasm divides, leaving you two daughter cells. And that's it for this slideshow. Okay, so I know this second half was probably a little bit trickier to grasp than the first half. Like I said, I really highly recommend watching the video links embedded within the PowerPoint. I think seeing the illustrations of things really can help. And your lab activities are all geared towards learning activities that should help you better understand some of these concepts for sure. So if anyone has any questions about any of the stuff in this chapter, please reach out to me via email. And I hope you guys have a good day.